Thank you to everyone joining us today for Cortic's webinar entitled Using AI to Optimize Process Development, a New Paradigm for Efficient PD. My name is Ramsey King, Marketing Lead here at Cortic, and I'll be hosting today's webinar. This is our 7th in a series of webinars that takes customers through the journey from operational data to actionable intelligence. If you haven't seen the previous webinars, you can find them on our LinkedIn page or our YouTube channel, where this will also reside after today's session. Joining me today are speakers Suresh Ramanan, Associate Principal from ZS Associates, Chris Demers, Principal Automation and Data Scientist from Catalent Pharma Solutions, and our very own Vinod Rodriguez, VP of Customer Success and Product Owner of Life Sciences here at Cortic AI. Cortic AI provides a powerful, open, and modular operational AI platform with industry-specific AI applications that accelerate time to value for our customers' Industry 4.0 initiatives. Our focus in today's webinar is how life science process development teams can leverage modern digital technologies with AI and machine learning to accelerate their product journey. We'll start with Suresh talking about the benefits of building out a digital strategy in process development. Next, Chris will talk about the challenges of the status quo in PD and provide insights on the value of a digital strategy from his experiences in the industry. To close, Vinod will talk about a better approach to PD and give a brief demo of Cortic's capabilities in this space. We'll wrap things up with a question and answer session, so feel free to post any questions in the chat or QA at any point during the webinar. With that, I'd like to turn it over to Suresh. Thank you.
Thanks, Ramsey. Thanks for taking time to hear us, everyone. My name is Suresh Ramanan. I'm an Associate Principal from ZS, and I'll be spending next 15 minutes to provide an overview on impact of digital transformation in the process development for pharma world. Before I venture, I'd like to set the context what PD means, process development means, and what digital means. I think this is very important to understand and have a common definition for both. Let me start with the process definition. It's all about designing the process and activities to be considered for developing upstream and downstream biologics for manufacturing. Upstream process development comprises of workflow from cell line development to a developed bioreactor process prior to the harvest and purification steps. The downstream process refers to the recovery and purification of target molecule after the upstream process. It includes harvest, purification, and viral clearance steps. A key challenge process development phases is to strike a balance between product quality with process performance and to accelerate innovation, speed, and robustness to support biotherapeutic protection. So as you see here, process development is not only about determining the critical process parameters, critical quality attributes, and spec limits, right? Those are all some 3 major aspects, but there are many things that process development pursue or drive the outcomes in terms of drug launches, which are really important, and the acceleration of those novel therapies are very critical. And obviously, the lead time has become a differentiating factor for the pharma companies during the tech transfer, and that will be really enabled by process development if we see it speed up the process. And another aspect is a speed to specification limit without wasting time on various quality multivariate analysis, and then speed to design is also very critical for outcome-driven challenges. The last but not the least, address regulatory needs and make sure the commercial manufacturing is set up for success to provide that outcome that is determined for process development. So with all these things, let me talk about what digital means. It's obviously a lot of companies drive digital, but it's all about providing ways of doing things to drive business value, enabling process for customer experience, and embrace right technology for business needs. So it's all about making sure we have the business value driven by process and adopting technology to provide that outcome, right? So before we go too deep into that, what drives digital adoption in PD, right? There are 4 or 5 things which comes to my mind. Number 1 is downstream process visibility and seamless integration of data. I think every organization likes to have that visibility end to end. That is more importantly to drive what is happening in the process. How can I make more informed decisions? And without a proper institutionalization of process knowledge in the organization, this might be a challenge. So having that one singularity of knowledge management in the organization becomes very critical. And couple of other aspects, faster and better product to market is very critical as we design the process upstream. So it helps to identify what is the right quality and what provides better reliability of products to the customers and patients to provide better experience for them. And most importantly, shortening the tech transfer time and make sure there is speed to manufacturing happens, if at all, even within the pharma or even within the CDMOs. It's very critical to speed up that particular process. And having the adoption of digital for 4 or 5 levers which we are showing up here will really enable the outcomes that we envision. So let me talk about what are the key things that digital strategy and technology platforms helps to drive process development, right? So the first step, having that dream to reality roadmap. This is how I call for easy understanding, and defining that North Star with the outcome of delivering exceptional patient experience and through a seamless process development of drug therapies. This North Star should connect with the enterprise-wide digital goals. For realizing this North Star, organizations should consider business and IT collaboration through a seamless operating model, data access, and cultural impacts. So that set the context for the levers that we are trying to drive using digital. Let me talk about 3 or 4 examples out of this, and then we can go to the next set of storyline. So digitalization of knowledge management and contextualization, which is very critical for both upstream and downstream process development. And there is a significant amount of intellectual capital being created by the experts, which unfortunately resides within their own ecosystem. Having an efficient knowledge management and contextualization can really enable the user to use the historical data and the knowledge of the process design and optimization, and obviously helps in reduction and redundancies in knowledge creation. Contextualization with comprehensive metadata can remove any dependency in humans for knowledge transfer. Standardization and knowledge creation can further be instrumental in tech transfer and knowledge traceability. When working with CMOs, this is very critical due to limited visibility into their operations. Just for example, learnings from the outcomes on one unit operation, such as API drawing for one program or an asset, can be used to design the process for another assets. Data and knowledge from one workflow could be leveraged to design and optimize the other workflows. For example, solubility measurements can be used to design a crystallization process and identify the right solvents for chemical synthesis of products to get the desired yields. So as of now, digitalization is very critical, and that really helps to build something called, which I normally call in many of our client transformation enablement, which is DIKW: data, information, knowledge, and wisdom. Most of the organizations are collecting data. How to use the data to make some informed information and intelligence, and then use that intelligence to drive knowledge and towards wisdom is a very critical framework that people can, or organizations can adopt. Let me focus on advanced analytics for process design and optimization. As we all know, data is a new oil. Pharma companies started leveraging advanced analytics such as machine learning models for property prediction and protocol design for unit operations, and many others. Also, as a result of digital transformation and connected data ecosystem, standardized and contextualized institutional data can be augmented with external knowledge to develop high fidelity ML models for property prediction. As the process design persona directly interacts with the data, empowering them through self-service analytic capabilities and computations in their workflows will only enhance their speed to delivery. Just for example, accurate small molecule API property prediction can significantly reduce the number of experiments needed for process design. AML models can be used for, number one, selecting the right solvent for yield optimization, and number two, developing a digital twin of unit operations to stimulate the impact for any changes in the process. The third one, which is really important, and many organizations are moving in that value chain, is automation for quality by design. Automation is a key lever in driving AI and digital transformation, which can lead to workflow automation and knowledge traceability, but also the ability to support multiple assets and programs in parallel. Lack of automation in knowledge capture and contextualization leads to missing information on important metadata. Workflow automation would help in standardization of knowledge capture and would simplify access to cross-functional teams for efficient knowledge transfer. Just for effective quality by design adoption, closed-loop AI capabilities can additionally be incorporated for advanced process monitoring. All of this could further ensure that process delivers the desired critical quality attributes, CMAs, and CPPs. And obviously, workflow automation and contextualization has the promise to reduce the time and resources needed to develop the control strategies. I'll give a couple of examples. Using high throughput experimentation integrated with PAT and automated data acquisitions, analytics, and documentation could save significant development time and also bring standardization in knowledge creation. One other example is process and workflow automation could additionally bring downstream benefits such as automated chemistry manufacturing controls content authoring by minimizing the time needed for transformation of control strategies in an appropriate module. So automation tech transfer documentation is also enabled through this type of digitization automation, which should be tremendous, helps in tremendous saving in time for data verification and would reduce risk of transcription errors, especially if the lab data can directly be populated in the templates. So there are multitude of, I would say, the strategy that can be used to drive digital in the process development scenario. So one other, but not, it's not last, but it's very important, which is change management. One size fits all does not work with digital transformation. And adopting a creative digital transformation for your organization that aligns with the culture, which will help to succeed and then go more distance than a shorter term success. Let me try to summarize what is the business impact of business or the digital adoption in process development. So transformation does not happen immediately. We all know that, and it requires a strategy and execution thinking. While the organization think of digital transformation, the duality always exists. There is a need to improve the current core and also develop new business models for exponential improvements. So it means that we need to run better as well as run faster while we are adopting digital. And there is a significant impact on interconnected intelligence for decision making, and then that drives faster time to market and improved quality outcomes, so on and so forth. By all means, we are not saying humans are going to be out of equation, but human in the loop, where the business of science and expertise matters, will drive much better outcomes for the clients and adoption of digital. And without the culture of innovation and without the fear of failure, I think adopting digital will take us to provide improved efficiency, enhanced outcome effectiveness, and enable better experience are the key levers that are driven for innovation and digital. Thank you for the time. And from now on, Chris will focus on sharing his experience in driving AI and digital transformation within Catalan. Thank you.
Thanks, Suresh. So I've had the fortunate opportunity to work for a couple different CDMOs, so I'm going to walk through kind of our current state. So as we're building towards some of the things that Suresh mentioned in terms of digital transformation in the PD lab, I've been able to see both current state and also play a role in our own digital strategy at Catalent, so I can kind of speak to where we're headed. So currently, the way that we treat new customers coming into Catalent and at other CDMOs is basically a blank slate. Doesn't matter if you've got an existing process or no process, we're probably going to take the same approach. We'll screen some media types, some process conditions, filters, columns, that sort of thing. And really what we're trying to do is strike some balance between what we know works or what folks at the company know works historically, and kind of our limited amount of time and resources. We can't spend an infinite amount of time making your process perfect. because usually we're one of the final steps before some product hits the market. So we have to move pretty quickly. And we kind of always assume that this design space stays the same from customer to customer, but in reality, the whole design space is probably changing from cell line to cell line. So we may not be anywhere near any of these maxima. We're probably testing some portion of the design space. And all this knowledge is just based on tribal knowledge, basically, and that really only gets you so far. And the reason for that is because we're dealing with biology. And biology is incredibly complex. And, I often say that we try to make a science out of it, but, we just, a lot of times we just don't really know what the underlying biological process is. And even if we did know, we are biologically incapable of understanding all of the complex relationships that happen, even in something like a bioreactor. There's so much going on, so much complex biology. I like to kind of tell a story. I used to work in stem cells, embryonic stem cells, and nowhere is this more true of this tribal knowledge than in stem cells. So there's this concept of these feeders that we just grow these stem cells on. And we don't really know what the feeders do. That all we know is that they prevent the stem cells from spontaneously differentiating. If you plate them out onto a hard surface, they're going to become bone. But if you keep them on these feeders, they stay embryonic stem cells, pluripotent. Well, some intrepid engineer came along and said, well, I wonder if it's just the fact that these feeders are soft, that they feel like other cells. And it turns out that's exactly what happened. So for so many years, we just kind of went along with, hey, let's just plate some feeders down and grow these stem cells. And then finally somebody figured out what the underlying biology is. So what we're proposing is, instead of relying on all of this tribal knowledge, what if we built out some knowledge database? And that's really only enabled by the digital transformation of our PD labs. So we're talking about things like machine data, things like molecular data, scale data. Did this process scale up correctly? Quality data. What's your product quality? Everything in a single location for all stakeholders to consume from. That's what our knowledge database is built out of. And I would argue that this approach is absolutely required for machine learning and artificial intelligence, because if you don't have everything in a single location, your very expensive data science teams are going to spend most of their time just cleansing, collecting, filtering data. And not only that, these initiatives aren't going to scale. They're going to, for the next project, they're going to have to do the same cleansing and filtering again. And I also want to kind of elaborate that it's more than just a data lake that we're thinking about here. It's really about the structure and the relationship of everything to each other. So I'm an upstreamer. So my example is an upstream one. So if I take a sample from a bioreactor and I walk it over to QC, or if I'm in the lab in analytical development, they are going to enter that data into some LIMS system or some electronic lab notebook. I want access to that data. I want that data to go back and aligned with my bioreactor data, because I want to know like what mass flow controllers were plugged into that bioreactor. When were they last calibrated? Who took that sample? What time of shift were they on? Was it the end of their shift? Were they trying to move fast? All the bioreactor data. How was the agitator? What were the gas flow profiles? Everything all in one kind of location. So it's all nicely contextualized. So for a CDMO, what would this capability enable? So let's say you come to us and it's just like two men in a molecule, right? You've got no process. You just have this breakthrough drug. Well, we could quickly compare to all of our historical data to suggest some design space. So if this is my, oversimplified principal component analysis of everything we've ever done, including all the molecular data and all the process data and scale data, and we plot your molecular data along with our previous runs, like here's your stuff. And we already know what the process was for all these other customers. And we also know, did these scale up correctly? So we can quickly hit the ground running and know what the design space is. Or even if you come to us with an existing process, we can quickly compare it with historical data with other similar molecules to predict the performance at scale, and one step further to suggest potential optimizations to your process that might increase titer or whatever we are trying to optimize around. At Catalent, we have a patient-first mentality. So for us, it's all about the patients. It means faster time to market. If we can leverage our historical data to expedite tech transfer timelines, then that means we can move quicker. And so for us, obviously, increased or shortened tech transfer timelines means more runs per year. And then for our clients or sponsors, it means decreased costs because, maybe because we know the design space, maybe we don't have to do quite as many runs, so that's decreased in raw materials utilization, or maybe just smaller batches because we've figured out a way to, leverage our historical data to increase the yield. And then, but for us, of course, the benefits go way beyond PD. As a CDMO, it's really all about manufacturing at the end of the day. And these two data silos often exist of lab data and manufacturing data. The problem is if we just rely on manufacturing data, our runs might be just in the single or low double digits. So from a machine learning perspective, that's a pretty small data set. But if you factor in then small scale data, one run on an Amber 250 could be, 24. If you do a couple of them in parallel, you could get 48 plus bioreactors really easily. And not only that, it's really our best space to explore the design space. So nowhere else in our facilities can we poke and prod the process to really push it to its limits. And like I said, even if we have all this data, it's going to be siloed away. A lot of times labs will use some, discrete lab system like electronic lab notebook, and it's not aligned with manufacturing data. It's not contextualized the same way. And so it's really difficult to leverage this data during the scale-up process. So if we were to have all this data together, maybe this is your batch evolution model and it's plotting along nicely, and then all of a sudden something has gone wrong, we can dig down and maybe a pump was turned on and is, maybe it's a base pump or something like that somebody accidentally left on. So not only can our data then catch this, but also we can say like, hey, we actually, somebody made this mistake and did this once in the lab, and it turns out that the product quality was fine. So we can quickly do a root cause. So in that sense, it really enables anybody to become an expert. So we don't just need our quote unquote A teams or people who have seen it all, been there, done that. Anybody can kind of look into this database and say that, get to the root cause quickly and efficiently. And really what we're providing the foundation for is what's called model predictive control or advanced predictive control. Currently, if we the way that we operate, we kind of assume that if we do the same thing over and over again, we're going to make the same product. And I don't mean the same product. Obviously, we're going to manufacture the same molecule, but the same profile, the same quality attributes, same titer. But at the end of the day, again, we're dealing with biology, and there's so many factors at play, more than we can even comprehend. It could be really subtle differences in media components or in the age of the cells. So instead, what if we controlled our process around some predicted outcome, something like titer or quality attributes? So it's more of a product-based control. Like we are predicting that, the titer is going to be X, but if we just, tweaked the temperature or did the pH shift on this day instead of that, then we could increase that, on the fly. Or that our product quality is starting to, maybe a charge variance is starting to slip, and if we tweak the process on the fly, then we can bring it back into control. And for a CDMO, all of these techniques, advanced process control, we're running multiple products, sometimes shorter runs at scale. So really, all of this hinges on linking in the lab data. Because again, that's our only space to build out our machine learning database. And not only that, whatever tool we deploy, we need it to seamlessly integrate with process analytical technologies, PAT technologies. And that's going to provide us with a lot of additional information about what's actually going on in our bioreactors. So, for example, if I just had, let's say, historian data, pi data, and I tried to make a model to predict the charge variance of a given product on day 14, it probably would do a pretty terrible job of actually predicting. But if I had a Raman probe in there and I had all of this data going up into a centralized location, and I asked it to predict the charge variant on day 14, it would probably, do a halfway decent job of doing that. So with that, I'll hand it over to Vinod to speak to the Quartic approach.
Thank you, Chris. So what Chris and Suresh really focused on are the challenges of implementing the system and what's required of these systems when we do want to implement them into a PD space. So what I'm going to briefly spend some time on is looking at the technical challenges of implementing this system. So essentially what we're looking at now is going through the workflow of implementing this system in a PD space that's not really designed to accommodate this. So ideally what we get out of this is two major pillars of challenges: the data and the infrastructure, and the complexity of analysis. The data and the infrastructure, we really look at multiple siloed equipment. As Chris mentioned, things are not meant to talk to each other. They're not set up in a clear automation manner. Equipment sometimes is transferred between labs as well. And of course, the data that these equipments are spitting out are sometimes in output files, sometimes streamed, come in many different formats. So the workflow here is to how to seamlessly integrate all these systems in a lab environment, in a PD space, so that I can not only collect information from quality databases, from historians, from file outputs, but have all this information contextualized as well under the context of a product, a campaign, or experiment. That's usually one of the major challenges that a lot of legacy labs face. Once we have this information, then comes the complexity of analysis itself. For example, if we look at simple DOEs in the DOE space, as Chris mentioned, the process expertise, the tribal knowledge, so to speak, is generally required to look at identifying critical parameters, looking at designing these experiments, identifying these factors, and what test points these factors need to be executed at and experimented at. And of course, the list of parameters grows as your experiment scales up as well. That just leads to essentially more time-intensive, labor-intensive, and money investments to execute these experiments. So if I want to fully explore my design space, I'm looking at a space that's going to require a lot of different experiments, a lot of different set points, a lot of different material attributes being captured, and then analyzing this data seamlessly in one integrated environment. So our current process and our current legacy environments don't have this infrastructure ready to be able to leverage all this data and leverage all this information. And that's the intent of our Quartic PD optimization tool, is to build out this infrastructure and then seamlessly integrate this to move into an optimization space. So for example, what essentially happens here is this Quartic's PD optimization tool combines SME knowledge with machine learning to define and explore a product design space while continuously recommending experimental settings as well. The application also serves as a knowledge management tool by essentially managing all the data coming out of these experiments, combining processes, quality, and PAT data to create a very holistic view of the PD space. So connecting to and integrating the lab equipment and these systems, digitally managing all this information, not just for one experiment, but a series of experiments, multiple runs, multiple molecules, including works that your colleagues are executing as well, is the aim of this, to gain visibility and then carry this knowledge about the design space, about the operating limits, the cliffs, the CPP to CQA relationships, to carry this knowledge through scale up and tech transfer as well. And again, the idea of accelerating your own experiment and development phase, but also accelerating the entire product's life cycle as well. So this is the kind of information, this is the kind of knowledge that's required that really becomes the foundation for QPD and CPB type programs. So with that in mind, with this context in mind of understanding these technical challenges of integrating data, of bringing in your experiments, bringing in your previous experiments, and then designing new molecules, what I'll do now is jump into our demo environment. Okay. So this is the Quartic environment, and what I want to do is just pretend to be a chemist essentially working in a small molecule lab. So what I want to do is look at how my lab is defined and contextualized in this environment. So I have many different process units here. I have a centrifugation, filtration, sterilization, and a fermentation unit with many different assets in these units as well. For the purposes of today's walkthrough, I'm going to focus on this test asset defined here in my fermentation unit. And the idea of building this context of a fermentation B13 process unit and individual assets and components within this process unit is to link those different siloed integrated data systems. So in this asset, again, of benchtop reactor class, and this can be customized and changed to every type of equipment we have in our labs. But the intent is to connect to these data sources that I have, whether they're in file data types, they're streaming data types, or simple one-off sensors like a PAT probe as well. So for this specific reactor, I have a PLC that's pulling in some information like my flow rate, my temperature, my pressure, my pH. And I have a LIMS connection that's pulling in some quick lab samples. I even have an IR probe connected to pull in raw spectral information. The idea here is that we have contextualized and connected and contextualized this information under the guise of an asset name. And we can do further contextualizations as we move into a manufacturing space under a product and recipe name. But for our focus, we're going to look at this asset context and then see how we can explore this asset context and leverage this asset context into a PD space or into a PD environment. So for that, I'm going to switch to a design of experiments, our PD tool here. The intent of this tool is to look at managing all those data, all those different experiments, campaigns, and products that I'm running, and then essentially also working as an optimization algorithm, as an optimization tool within those experiments as well. So I have a few different projects that I'm going through here. So for example, let's look at this ampicillin project that I'm created. You see I have a project in process here. And essentially what this is giving me is a quick summary of everything I've done for this process. So if I was to start, if I was to resume this experiment, I'm going to connect to that, excuse me, to the Spectral Test Canada asset that I defined in my environment. And I have a few PLC and watchdog timers here just to make sure that I keep that connection. So I'm going to take control of this asset and essentially start to build a series of experiments to control this reactor to perform whatever it is I need to perform. Now, in this environment, the way we are building this optimization workflow with this reactor is by taking an IR sample of our target product, identifying the IR peaks within that target product, identifying the product peaks of interest, and then any peaks we may consider impurities with the goal of maximizing my product peak and potentially minimizing my impurity peaks again. So I've manually selected this here, but this doesn't have to be a PAT spectral output. This can be any measurement, as Chris mentioned, a titer measurement, a quality sample measurement, or even an online sensor measurement coming off your reactor or your equipment as well. The goal in this specific optimization workflow is to look at connecting to that asset, pulling in that pressure, the flow rate, the temperature, the controllable parameters, and essentially having Cortex suggest set points and continuously recommend set points that could lead to this optimization of this peak and the minimization of these impurity peaks here. So this is the control. This is the full closed loop control that we've simulated with a digital twin. So we have a high fidelity model that can simulate this process, and we can run these processes very quickly in our demo environment here. But ideally, this would be connected to your automation systems, controlling and setting the reactor set points, or this can also be done completely offline as well. For my purposes here, you see that I've completed about 11 different runs. And out of these 11 runs, I'm going to add 2 more runs of experimentation, asking Cortex to suggest set points between 1 and 5 values here. And then essentially resume this optimization workflow of suggesting set points and trying to maximize this output here. This workflow is all going to be captured under the process of this ampicillin project, as well as every run under this, every experiment under this will also be captured under this ampicillin project. We're keeping track of all the different features, the set points of these features, the start and stop times, as well as the output measurements coming from the PAT measurements themselves. Again, it does not have to be a PAT measurement. It can be an offline lab sample that may take a couple of hours to a couple of days to turn around. So this is working in real time, but it can work in near real time, in continuous, or completely offline as well. So what's going to happen here is Cortex will suggest set point changes to the reactor. And again, our digital twin, our high fidelity mechanistic model here is going to implement these changes, get the output out, and we don't have a simulated IR probe. So we're just going to get similar IR out readings throughout this process. But essentially what happens is we can see for every iteration how this IR profile changes, or if it's not, again, PAT, how your titer profile changes or how any CQA measurement changes throughout this process. Because this profile isn't changing in a simulated environment, we don't see the feature influence of each of these features. But ideally, the workflow would look like identifying these features, identifying which ones are contributing to the changes and the objective of maximizing my product peak here, and essentially identifying which features influences and the percentage of those feature influences. This is that entire design space. Once I run through my experiments and my different runs here, I want to identify what are my ideal set points for these reactors. And what we've set up here is a continuous workflow. So looking at a set point that's almost set at one time and run throughout. In the same way, you could have set points that follow a specific recipe that change every hour. All that is completely applicable and customizable through the UI as well. In terms of our demo purposes, we just kept it as a simple continuous process here. Once I have this information, what we can then do is look at essentially saving this project out and continuing this project in a campaign now. So I'll switch from our design of experiments view now into our campaign manager, and in this case, our experiment manager. And we can start out essentially building out a campaign, for example, qualification or for scaling up or testing out to see if these set points are repeatable. If we can manage all this data, if we can draw from the previous amoxicillin test data and run this set points now in a less of a lab environment, more of a manufacturing environment, or just to look at validation and consistency of my runs. So let me just do a quick project here. And I'll choose one of the fill-in material IDs I have, but essentially this could be any number of parameters, any number of set points, any number of features that result in my project itself. So in this case, I'm going to continue with this project. Here, I'm going to start by saying, let's just look at these features. Again, this specific process requires these inputs, but it can be completely malleable and changed and customized to any process. And once I've started my campaign, I can look at all the different runs for this campaign itself. So here is again, a few input parameters. Set out if I want to write this output back to a specific timer, back to a historian. Once I take over control for this PLC, now I can start essentially running these experiments in real time, running these workflows in real time, and capturing the design space, capturing the operation space in real time as well. So now essentially I've taken the best optimal set points for my runs, and I'm going to run, in this case, going to run multiple different runs to control my reactor, measure its output, see what that output is, validate the output itself, if it works in reaching my objective, and if not, suggest more change points and set points if needed as well. This entire workflow, again, is automated to show a closed loop control scenario in a PD lab, but it can also be performed completely in an offline manner as well. So instead of connecting data and connecting equipment, which may be difficult in legacy environments, you can upload data via CSV, you can do periodic measurements. And in this case, as my run simulation executes, it'll start to fill in this information and fill in these outputs, pulling from these different PAT equipment, from quality databases, from historians, from control systems, and essentially building out this PD space. This demo is also focused on one piece of equipment. So as this finishes, you'll see the data start to populate here. It's only focused on one piece of equipment, but we also can link multiple pieces of equipment to look at an entire process unit. We can link multiple process units to form a train. And if you want to build an entire upstream and downstream suite, that also can be accomplished. The idea here is to look at data and integration, and then use that integrated environment to run as much complex analysis as required. So I'll let this run in the background, and I can open the floor now for any questions. And I know I went through a lot of different things in the demonstration here, but I can open the floor to any questions from the audience here. Okay, this looks like we have too much questions here, so I'll stop sharing my screen. Ramsey, give it over to you.
Sounds good. All right. So, as first of all, thank you to all of the presenters today. That does conclude the content of the webinar. I did want to open the floor up to general questions for any of our presenters, and it looks like we already have the first one in. So what I'll do is I'll read these questions off, and then the panelists can respond accordingly. So the first question we have is, you mentioned that this works with a digital twin. Can you explain that more? Vinod, I believe this question is geared towards you.
Yes. So in this demonstration, what we've done, instead of connecting to an actual piece of automated equipment, what we've done is connected to a high fidelity digital twin that we had a mechanistic model running in our backend here. So if you have a digital twin of your process, a first principle based model of your process, we can essentially connect this optimization workflow to that digital twin and run in silico modeling. So essentially running different set points to be able to identify what that process looks like, how it reacts to these set points. And then that way it saves us on running actual experiments in practice. We've also worked with customers and with academics to develop these digital twins. It does take some information and a lot of tribal and SME knowledge, but we can work to help to develop these digital twins if required as well. So for example, in our environment, we have one running of a penicillin process. We have one running of an AMAP process, as well as a food and beverage example of a brewery process as well. But essentially, what this optimization demo is doing is connected to a digital twin just to simulate some quick control back and forth. And that can be done either on premise, that can be done completely offline. It's completely up to you.
Thank you. Are there any other questions? So here's another question that just came in. How many experiments do I need to run to build a model? Can I use data that I've stored somewhere to get started? Vinod, I believe this is also for you.
Yep, that's a good question. So our optimization approach is touting that we want to be able to essentially leverage the ML algorithm to reach a steady state or reach your optimal state quicker and more accurately and confidently than a DOE would. So essentially, what we can, yes, as a short answer, you can use historical data and build historical information from your previous runs as a CDMO or from your previous lab runs to predict a new product or to optimize a new product that's going to be connected. That can absolutely be accomplished.
Fantastic. This is a question that says, can you please share the slides? I'm assuming this means after the webinar, and if that is the case, then we will be posting a video, the recording of this webinar. And if I'm not sure if we'll be sharing the actual slides themselves, but we can definitely see what we can do there. Is there any other questions? Okay. So with that, we first want to thank everybody who joined today. I especially want to thank Suresh, Vinod, and Chris for presenting. What we like to say is that, all of our like I said, this is the 7th installment of our webinar series. All of the prior webinars are live on YouTube and our LinkedIn, where this will reside, in a day or so after we're done today. I also would recommend keeping an eye out for future webinars that we'll be hosting on a variety of topics. So with that, I'd like to wish everybody a great rest of their day and a sincere thank you for joining. And if you want to contact us to have a more in-depth conversation about how we can work with your specific processes, I implore you to reach out to me directly. My email is ramsey@quartic.ai, or any member of our team, and we'd be happy to set up a personal discussion where we can really dive into the details of what your processes are, what your needs are, and how we may be able to address them. So with that, I wish everybody a fantastic rest of your day, and our deepest gratitude for joining, and we look forward to continuing the conversation down the road. Thank you, and goodbye.